Again, welcome to the Innovative Genomics Institute Open House. We're pleased to see such an amazing turnout. This is really incredible. We'd like to begin with the presentation part of the open house. So the IGI leadership, who you'll meet over the course of the next presentations, can tell you about the specific research programs and then also other activities around which the IGI is building a very multidisciplinary and multidimensional organization. So while it takes many, many people to run an endeavor such as this, there's definitely some key people who are really the key leaders in this project. And most of these individuals you'll be hearing from over the next half hour or so. And without any further delay, I'm going to turn it over to Jennifer Doudna, who will welcome you and get us going. Thank you very much.
Well, good afternoon, everyone. Thank you, Susan. It's a really exciting day for us here at the Innovative Genomics Institute. We are delighted to see so many people here. Is this actually on? Yeah, it's on?
Yes.
Yeah, if you hold it. Yeah, hold it closer. Good. And so what I wanted to do very briefly is to tell you a little bit about the history of this institute and what we're trying to do. I want to introduce you to my co-directors here, and I want to turn the floor over to them to tell you about the exciting research programs that we have going on. We invite all of you to learn more about us, get involved in any ways that you are so inclined. This institute actually started back in 2013, believe it or not. So it was an idea that came about through conversations I was having with my colleagues both here at Berkeley and at UCSF about the opportunities to use gene editing in different ways to solve real-world problems. We could see incredible opportunities in that area. Our former dean, Steve Martin, was convinced that this would be something that Berkeley should really take the lead on. And so we were able to establish an initiative at the time, and we had some space in the Li Ka-shing building across the street initially. And then, in partnership with my close friend and colleague, Jonathan Weissman from UCSF, we teamed up to look for someone who could come in and lead the scientific effort at the Innovative Genomics Initiative at the time. And we couldn't think of a better person than Jacob Korn. And so Jacob Korn at the time was running a research team at Genentech, and we were fortunate to recruit him here to be the scientific director of the IGI. And you'll hear from him very shortly. He's built an incredible team. We have exciting collaborations going on with Chori and with our colleagues across the bay at UCSF in biomedicine, and also in sort of furthering the technology development. And then from there, we could see that there were great opportunities in agriculture, and that led to the recruitment of Brian Staskowitz, a colleague here at Berkeley, who's running the plant genomics team here at the IGI, and you'll hear from him shortly. And then as we were developing the biomedical and sort of expanding the biomedical goals of the IGI, we recognized that there would be a really exciting way that we could develop a true pathway to the clinic for this technology. And to do that, we needed to partner with our clinical colleagues across the bay. And that led to the involvement of Bruce Conklin, who you'll hear from shortly, who runs a lab at the Gladstone Institute at UCSF and is a leader in genomic medicine and is helping us to craft the vision for how we're going to create a real pipeline for using gene editing to cure genetic disease. And then finally, we've of course always wanted to go back to our roots, which is really fundamental science, and asking how can we use fundamental questions about biology and in particular microbiology to drive future technologies. And that led to Jill Banfield, a professor here at UC Berkeley, joining the team to run the Microbial part of the Innovative Genomics Institute, and you'll hear from her shortly as well. So without further ado, I'm going to turn the floor over to Jacob.
Thanks, Jennifer. So, let's see if I know this. Okay, so I just wanted to start off. Things have been moving crazy fast, and so sometimes I also lose track of what happened. So I just wanted to give a really quick history of the IGI from sort of the point of view of biomedicine. As Jennifer said, she and Jonathan and Mike recruited me in 2014. This is the way the lab looked when we first got started in Li-Ka-shing. It was 2 benches in Li-Ka-shing. And at the same time, we launched an IGI affiliates program in which we reached out to people that sort of had complementary interests in genome editing and genome regulation. And through really great support and having really fantastic colleagues, it was actually only just about a year later that the first IGI affiliated paper was published. Very soon after that, an IGI affiliate reported preclinical editing in primary human T cells. Soon after that, other members of the IGI reported preclinical editing in human HSCs. And this has all really gone very quickly, so that now, at this point, together with colleagues at Children's Hospital Oakland and UCLA, we're building up towards pre-IND studies, that is, clinical trials, in gene editing to cure sickle cell disease. And so that's a little bit crazy, right? The Institute gets started in 2014, and 2-3 years later, we're looking forward to maybe start clinical trials. So I think that's one of the amazing things about this community, is that everybody is really excited to work with each other. And really, it's sort of amazing how quickly this technology can move. Just to give you another sense of how that works from sort of a more academic publishing standpoint, these are IGI affiliated papers. So the Institute gets started in 2014. Within a year, there are about 5 or 6 papers published with IGI affiliations, and you can see that things are just growing from there. So it's been a really fun ride, and I think that this has also been sort of evidenced by complementary funding that's come in. As more and more things happen within the IGI, it's led to more and more interest in IGI and in genome editing, and I think that's one of the fun things. But I wanted to point out one extra thing, which is efforts that we're trying to do. IGI is not just about IGI. We're part of Berkeley, we're part of UCSF, and we really want to make sure that we give back in a way that helps everybody. Genome editing is this crazy thing, and it's hard to keep track of how quickly things are going. So one of the things we do is we try to create tools that will help other people accomplish their own research goals. So for example, with support from UC Berkeley itself, through the Berkeley BFRF, there are Cas9 ribonucleoprotein complexes that are available for free for anyone on campus. You can go down to something called the macro lab and just get them. All you have to do is write a one-paragraph sentence, little one paragraph about what you want to do with them. You can use them to edit cells. BFRF also supported these things called high-throughput nucleofectors. You can edit 96 cells at a time with different conditions. They're available for free. You can go out, you can use them tomorrow if you'd like. Similarly, Berkeley also has CRISPR cell lines. You don't have to make your own. You can just get them for free from Berkeley and UCSF. Jonathan Weissman has made a lot of his libraries available, again, for free. The Berkeley viral packaging facility not just takes vectors, but also makes the virus themselves available. You don't have to be doing a lot of this work yourself. We try to make it as easy as possible. We also have really tried to support campus in terms of physical facilities available. So there are cell sorters, cryo-EMs, things like that we think are really going to make a difference in people's research. And so we not only try to make IGI strong, but we also try to make the facilities available at Berkeley as strong as possible. And so these are just lists of some of the facilities at Berkeley and UCSF where we've tried to put in significant resources, either genome editing or regulation resources or physical resources, to try to change the way people are thinking about their own experiments and think about new ways that they can do their own research. So I'm just going to spend a couple of minutes talking about what is it that we do in biomedicine. In general, we're trying to think of ways that we can use Cas9 as a scalpel to cure genetic diseases. But that means that we need to do some really fundamental research first, because genome editing is just the scissors, and how we actually cure diseases is up to the cells themselves. And it turns out that process is not really very well understood. So part of what IGI does on the biomedicine side is try to understand how DNA repair works, what happens after the scissors go in, how does repair actually happen, how does that happen in different cells, how do we figure out ways to measure off-targets, to take care of off-targets, how do we use these as a research tool to figure out how certain diseases get caused, and then at the way down the road in the clinic, how do we actually treat genetic diseases? There are several projects that have recently been announced that we're helping support. And this is in campuses from UCSF to Stanford to Berkeley. I'm not going to go ahead and read these slides, but we're interested in everything from using genome editing and regulation tools to understand basic mechanisms of disease, to create new tools to treat genetic disease, and then all the way down to actually deploying those in ways that could be used in the clinic. So some of these have been pointed out as having posters, so I encourage you to really take a look for any of these where there are posters available. I'm going to pass it over now to Brian Statzkowitz, who's going to talk about efforts in agricultural.
So it's quite evident that applications in agricultural biotechnology are going to really take off using the genome editing technologies. And we really envisage genome editing technologies in plants as really the next kind of level of advanced plant breeding, where we can start with elite genetic lines of plants and then we can add particular traits to them. So basically, what we're interested in doing is, as it says right here, as it promises to grow food crops sustainably by reducing our reliance on pesticides or other farm inputs. And the IGI strives to realize this potential to benefit both the developed and developing world. So we kind of put it into kind of 4 major areas that we are working in. We're interested in food safety and nutrition. Well, I'll give you a couple examples of that. We're interested in developing plants that are resistant to pests and pathogens, because I think one of the major challenges in agriculture, as we know right now, we have 7.9 billion people. In 2050, we're going to have 9.8 billion people. We need to produce 70% more food on the same amount of land, and we have to do it by reducing farm inputs. We have to do it in an ecologically sustainable fashion. So that's a challenge that I think genome editing can address, and hopefully we're going to be a leader in this particular area. And of course, we have the challenge of climate change, and also developing technologies in plants are very important to go forward. So kind of our current efforts right here are, as I said, develop crops that are safer, healthier, and more nutritious to address human disease and malnutrition. I'll give you a couple examples of that. There are certain plants like cassava, which produces the 3rd most calories in the world for the developing world. They often produce cyanide, so we're developing technologies to actually eliminate that in conjunction with Dan Rockshaw's lab here at UC Berkeley. There are cacao plants that we're interested in that actually have advanced levels of cadmium that we're trying to reduce in that also. And they're also starting a new collaboration with Beth Satley at Stanford University, where she's working on engineering broccoli and other crucifers for anti-cancer components. So it's really quite exciting, some of the varied programs that we're doing. We're developing high throughput, precise genome editing technologies, and we're doing it in crops like wheat, rice, corn, cassava, cacao, potato. Those are only a few of the examples. And to do that, we had to really establish, one of the first things we did here was to establish a first-rate plant transformation facility. And you'll be able to see that over here in the hall over here, where Myong Jae Chow is the plant transformation director, 25 years experience. We're very lucky to recruit him, plus he's an excellent technician. So we have a very important plant tissue culture transformation facility. But we're also developing ways to deliver reagents such as RNPs and other DNAs into plant cells. And we want to deploy these genome editing technologies in major crops to increase yield and as I said, in a sustainable manner. Now, once we do this, our long-term vision is to collaborate with these institutes called the CGIR, which is this large conglomerate of not-for-profit institutes around the world, and things like CIMMIT, which is in Mexico, AIRI, which is in the Philippines, CIAT, which is in Cali, Colombia, and CIP, which is in Lima, Peru. These people develop germplasm that gets distributed. So we'd like to establish relationships with these particular institutes to be able to disseminate the technology we develop here. This will be the way that we're going to put our stuff out into the field. Okay? Again, we're not going to, these are actually some of the projects, so I should also mention about several months ago we had a call for proposals. So we know we're here at Berkeley, but we also realize that UC Davis is a major agricultural center. So we have a lot of colleagues there, so we had a call for proposals, and so we're funding various proposals at Berkeley, Davis, and also LBL up on the Hill. And so these list some of the projects that we're doing, projects that we're currently doing here. We also have some very interesting projects, I think, with, well, for instance, Marquita Landry, who's in the Department of Chemical Biomolecular Engineering, where she's actually functionalizing carbon nanotubes to actually deliver nucleic acids into plant cells. This is very exciting. She's got some great results. And also Niran Murthy here, who's actually in the bioengineering department, and he's developed some very nice gold nanoparticle delivery of RNPs into cells. So I'm not going to really go through and read these particular, but I should just mention you can see that here, but there's a lot of posters that are out in the hallway that one can go see. I also invite you to come visit the plant transformation facility, which is just beyond the door right here. Go over here, take a right. I think it's a great facility. And I'll stop there and hand it over to Jill.
Good afternoon, everyone. So I guess you all know that microbes run the world. They also invented most, if not nearly all of our drugs, and they happen to have had one other remarkable achievement of note today, and that is that they invented CRISPR-Cas systems. So the objective of the Microbiology Initiative is in large part centered on studying microbial communities, including all of their various members. Historically, microbiology has been studied through growing organisms in pure culture in the laboratory. But this means that we've been missing the biology of 99.9 something % of all life on Earth. The situation changed dramatically in the last few years as methods to reconstruct genomes from uncultivated organisms came into play. As a consequence of this approach, we took a pause last year and we generated a picture of the tree of life that included both uncultivated and uncultivated organisms. I draw your attention to the simplified version on the slide, and I want to point out two things. First of all those red dots, which I hope you can see, indicate lineages for which there is no cultivated isolate. In other words, nothing from that whole major branch of the tree can be grown in the lab. I draw your attention to the right-hand side of this diagram, where we have basically an entire radiation within domain bacteria that is made up of these organisms. The main focus I want to talk about today is microbes in microbial communities, and I want to point out that the reason these organisms have remained outside of the scope of study is probably because they interact with other organisms, and so they cannot be easily grown by themselves. As a consequence of this, I suspect that many of the genes and pathways in these organisms are associated with interaction with other organisms, and those interactions could, of course, involve molecules and pathways to kill or manipulate cells and their surroundings to acquire basic resources. One of our approaches to the study of the vast number of genes and pathways that are turning up in these organisms that have no known function is to generate protein clusters or protein families, so grouping together proteins that have similar inferred function and similar characteristics in terms of their sequences. In other words, we're trying to define the protein family universe and then map those protein families onto the phylogeny. And what we're seeing is that there are emerging groups of proteins, including many, many, many groups of proteins of unknown function, that will be targets for further analysis. A particularly interesting group of molecules are secondary metabolites, which are generated by microbes in part to affect or even kill other organisms. In some of the work ongoing at the present time, we're studying soil microbes, and this diagram illustrates just one subset of those, and we're finding that they're making a huge variety of small molecules, and many of them are probably antibiotics. And the notable thing here is because we're studying these organisms with genomes, and they haven't been cultivated, we're finding very novel things that have not seen before. So one obvious direction is to pursue these genes and clusters to try and identify the molecules and to test for their functions. Along a similar line, some of the work that's being pursued through the IGI involves the search for new CRISPR-Cas systems. And I'm just going to say briefly that this search has been successful, and this year was published a study, Cas-X, Cas-Y, and Archaeo Cas9, in collaboration with the Dellner Lab. Another component that comes to mind is the need to be able to study organisms without growing them and to use genetic methods to determine what the genes and pathways are doing. And so an important objective for research on the campus and associated with IGI is to develop methods that allow us to do two things. One, to eliminate organisms from microbial communities so that we can probe interactions. And the second, to affect the functions of genes, maybe knock them out, add them, change their regulation, so as to test hypotheses about the function of those genes, for example, in interactions. So I'm going to conclude with just a simple slide that presents the list of papers that are being posters are presented today, and invite you to participate in discussions at the posters. So thank you.
Okay, Ross is in. Okay, let me introduce Ross Wilson. So Ross, just a very brief intro to Ross. So Ross is running a group here at the IGI, And he's interested in technology development and in particular how we can deliver gene editing molecules into different kinds of cells. And he'll tell you a little bit about what he's doing with that.
Thanks. All right, hi everyone. So yeah, it seems a little bit maybe silly to separate technology off into its own category because I think technology really underlies a lot of the things we're doing here, and you know the entire genome editing revolution. Really started when we discovered one key piece of technology. Cas9 really set things off. So what we're doing with technology development at the IGI is really trying to, as Jill mentioned, discover new analogs of Cas9 that can have different and beneficial properties, and also to take the tools that we have and make them work a little bit better. So, and another thing we want to do is find ways to take our existing technology and ask new questions that'll let us understand biology a lot better. So I just bring up this really classic slide to talk about two key things. One is that the traditional knockout that we do with Cas9, which is seen on the left here, it works really, really well, but sometimes it can work a little bit too well, and if Cas9 spends too much time trying to cut, it can make these off-target effects. So some of what we're trying to do is find ways to prevent Cas9 from cutting stuff it's not supposed to. And then another thing that we haven't figured out too well yet is how to make this pathway on the right really effective. So inserting a new piece of DNA to carry out a DNA correction through a process known as homologous recombination, it's been shown that it's really difficult to make this work efficiently. So we're also trying to improve this pathway and sort of steer the cell's repair towards the right. And that'll give us a really, really powerful editing tool. And the thing that my lab is sort of most focused on is how to do therapeutic genome editing delivery. So right now, genome editing is working really, really well in the Petri dish, but getting this to work inside of a patient is a totally different story. So getting these genome editing enzymes into cells inside an organism is very difficult because cells are very good at defending themselves against this sort of thing. So we have to sort of use whatever tools we can to perform corrections in the place that we need to. So I think a lot of the earliest successes are going to be through a technology called ex vivo therapeutics, which is where you take cells out of the patient, perform editing in a laboratory type setting, and then take those corrected cells and return them to the patient in a process known as autologous transplantation. So this is working pretty well right now, but the sort of preferable thing we would like to imagine is a scenario where you can sort of just have an injection and genome editing will take place inside your body. So the simplest way to do this is something called sort of systemic in vivo genome editing. But there are a lot of advantages to doing a targeted genome edit. So basically creating an enzyme or a delivery mechanism that would send the enzyme just to the cells or tissues or organs that are in need of correction. So this would probably lower your risks and decrease the amount of genome editing therapeutics that you would have to use. So my lab is really interested in sort of going after this pie in the sky challenge of having a sort of injectable in vivo therapeutic that will just go right to the organ in need of correction and do its job, and then leave you all fixed up. So like I said, my lab is interested in this question, and we're collaborating with Alex Marson at UCSF to find ways to correct hematopoietic stem cells, hematopoietic cells like progenitor cells, T cells, and essentially find ways to correct blood diseases. This could enable sort of like immunotherapies and a lot of really powerful stuff. Another lab that's working on delivery technology is Rich Micklemore's lab at Davis. And they're collaborating with the GUO lab to find new ways to deliver genome editing enzymes to plants. So a lot of plants are pretty easy to edit right now, but there are a few really difficult plants to edit, such as legumes. So they're going to use stuff such as microbombardment and nanoneedles to find ways to get these enzymes into the plants that are really tricky to edit right now. So a lot of the other technology projects are working on a broad diversity of problems. The Corn Lab is really a world leader in trying to find out how to promote homology-directed repair to do a corrective edit to a faulty gene. The Duber lab is finding ways to create a CRISPR-guided DNA polymerase, and they want to create a sort of mutation engine inside a cell. And so this can do, this is basically useful because it can promote continuous directed evolution, which is a way that we can use sort of the fast forward button to push evolution into the future and find a lot of new interesting phenotypes inside organisms. The Karpin lab is interested in finding out how the structure of DNA, whether it's scrunched up or spread out, and whether manipulating those states can improve genome editing and potentially steer repair towards homology-directed recombination. And John Marshall is going to be looking at gene drives. So this is the technology that we're interested in using to control phenotypes in animal populations in the wild. And John is going to be using some models to predict how these gene drives might actually work in the real world. Yeah, and the McManus lab is going to develop some new technologies to find ways to make lots of mutations to mice to find the relationships between genes and sort of uncover these complex genetic architectures. Ben Oakes is going to continue his work on finding ways to engineer Cas9, finding ways to make it into a sort of switchable enzyme, because like I said, if Cas9 is cutting for too long, it can cause problems actually. So Ben Oakes' work is focused on creating a switchable Cas9. And finally, David Siegel is creating new tools, molecular machines to report on edits that are happening to the genome, the epigenome, and the transcriptome. So this is basically going to be a really powerful research tool to see in real time how these different parts of the cell are being modified. Okay, so with that, I'll turn it over to Susan Jenkins, the managing director of IGI, I think.
Yeah, you have something?
I was going to talk about the social sciences.
Yeah, why don't you do that? Yeah.
Cool. Okay, everyone, Susan Jenkins.
So as you can see, there's already a wide variety of projects, different areas of research underway. And did I go, whoops, the wrong way, sorry about that. So we also have another area of research which is equally important to the great scientific research that's going on, and that's in issues that will impact us sort of in a societal, a socioeconomic way. And that's with on the human side with ethics, with treatments, and then also on the plant side with GMOs and environmental impacts of things that we might want to change. So we're really trying, we're starting some new actual research projects under this umbrella of the IGI. And these will cover areas listed there, as I already said: ethics, responsible use, policy, law, regulation. So we're looking at, at the federal level, FDA, USDA, EPA. Where does, how can we engage with these institutes or different level of, either, government level or at state or federal level, to make sure that we're proceeding in the right way in these different areas. So this area actually started before the Institute was the Institute, when it was the initiative, and Jennifer and other leaders have been very vocal in making sure that the scientists have taken a step back and have been very careful. If we really want to recognize or the promise of this technology, we need to do it in a very responsible way. So back in January 2015, IGI leadership was instrumental in organizing a meeting that took place in Napa on the called the Forum on Bioethics. And then as a result of that, the top scientific societies around the world convened the International Summit on Human Gene Editing, and there have been subsequent activities along those lines as well, and a number of reports published on how we should be proceeding or not. We've also, here at the IGI, in collaboration with other groups, have participated in a couple of activities. There was a series, or actually we're partway through a series of 3 talks, The Challenge and Opportunity of Gene Editing, a project for reflection, deliberation, and education. These workshops are being held, 2 of them have been held already. The final one will take place in January. You can learn more about that, I think at Megan's table, or talk to people in the IGI, it's on our website. And then we were instrumental in a recent event held here on the campus called CRISPRCon, which was really looking, bringing together industry, academia, and agencies to look at the science society and the future of gene editing. So the goal there was to try and bring different stakeholders together so that we can really figure out how to have the right conversation moving forward. And so we recognize these areas are extremely important, and at least at this point we have 3 projects that we'll be starting now. All 3 of them actually will have posters up, and the posters will actually be in this room. So when we clear out of here, this room will be turned over. A few posters are on the walls. Those are from the plant transformation facility. So posters will be in here, and I strongly encourage you to reach out and out of your disciplinary safety zone and talk to others who are working in areas that you would find interesting. And the other thing I'll add is at the bottom of the posters, there's actually a section for others to comment on. There's some Post-it notes. So if you want to engage with somebody, if you have ideas, you can leave a note. You can ask them to contact you or provide them your contact information. Because this is really about building a community. It's not about remaining in your stovepipe. It's reaching out across the different areas of research. And so I think this event is one way that we're really trying to try and make that happen. I'll mention a few more things about the different programs and opportunities that we've initiated so far. We really are looking forward to interacting with the broad campus community here at Davis and Stanford, SF, LBL, and beyond. We've launched the faculty and postdoctoral programs and recruited for those 2 programs over just during the last year. We had we provided research funding through an RFP. We are interested in developing additional collaborations, either by joint submission of proposals external to this campus or just use of the facilities some of the core facilities that we have in place. And there will be a lot of other events and activities going on, and a lot of those are being developed with our education outreach group, Megan Hochstrasser and Kevin Doxson and Lee Witkowski, who are around here today, and I encourage you to seek them out and talk to them as well. Last slide here for me is just to highlight the postdoc and faculty programs that we launched just this last year. We feel like it's great if we do this stuff at the bench, but if it doesn't transition out into the real world, it's not going to have the impact that we would like. And so a key program we started was the Entrepreneurial Fellows postdoctoral program, and the 2 first recipients, Alex Schultink and Ben Oakes, also have posters here that you can take a look at. Alex is in the plant biology space, Ben is in the more biomed technology space. And then the second program we launched was the Shirley K. Kersey Faculty Scholars Program. And the first scholar is Roberto Zonku. He's also here, has his poster, and would be happy to share with all of you the research that he's doing. The Faculty Scholars Program we'll be starting another call just after the start of the year. So we plan to bring in another scholar for the remaining 4 years of that program. And I think I'm around, I can answer questions later. One other thing I want to mention, so on these tables out here, there is butcher paper on the top. And at the center of the table is a question or asking for your input into something. If you could CRISPR something, what would it be? Invoke your inner scientist, doodle something that gives us ideas or inspiration. There's a table that asks, what do you think the IGI should try and do in the next 5 years? So we are looking for feedback from the community, and so if you go out and wander around the tables, pick up a pen, and try and help us by adding some comments to the tables. So, okay, so I'm going to turn it over now to Megan to talk about education and outreach, and that's the last section, then moving on to.
Thanks, Susan. So I'm the last speaker, so I'll try to be brief. My name is Megan Hochstrasser. I'm the Science Communications Manager for the IGI, and I really work pretty closely with Kevin Doxson and Dee Witkowski as well, so it's not just me. And I've been working on our education and outreach efforts. So we're very committed to sort of responding to the demand that the public has right now for scientific knowledge about gene editing. I think it's really popular, it's in the news all the time, and I think we have a great opportunity here to use our platform to get people excited about science and provide them the information that they're looking for. And so to do this, we have a few different approaches. We have online content, we have in-person events, and I'll just give you a little overview here of some of the things we're working on, but I'll also have a table up at the front later, so if you want to come talk more about that stuff, feel free. So the first thing I want to mention is a program called Ask a Scientist, and Ariana Hirsch will be at the table later also talking about this. She's one of our more enthusiastic volunteers, and she has a lot of fun doing this. So basically this program is something where people can just Google Ask a Scientist, go on our website, and get connected with a scientist from the IGI, or we have a few people outside of the IGI that do it too, and ask all their questions directly to the people that are doing the work. So we sometimes get questions that's just like, I'm a girl and I want to do science later, can I talk to a woman scientist? Or we'll get someone that has really specific questions about gene drives or something like that. So we're really looking for volunteers for that, so if anyone's interested, come talk to me later, or email me. And in addition to that, which is available online, we're trying to develop more online resources and make more content, and I've taken a snippet here from this whiteboard video that we had a high school summer intern make, where he explained what the PAM is, the protospacer adjacent motif, which is something that is kind of hard to explain and complicated. So I thought he did a really good job, and you can check that out on our YouTube channel, especially if you're streaming right now at home watching this on there. And so in addition, we're working on a whole bunch of content that should come out soon. I will give you a preview at our table of our CRISPR edited organism gallery. So I've been trying to keep track of every single species that's been edited with CRISPR so far and make it kind of nice and visual for lay people to look at, but then also include links to PubMed, so scientists can use this as a resource as well. And then the IGI blog, I should say, it's open for submissions if you're in the IGI. Even if you're not in the IGI, you want to use our platform to talk about something that's interesting. We've been putting up a bunch of stuff lately. We had some interesting posts from Mark DeWitt and Jacob Kornslad who talked about progress towards curing sickle cell disease, and then we had a patient with sickle cell disease who talked about the historic mistreatment of minority communities and how that influences how those communities look at these sorts of technologies. So I think it could grow even more than that, and I would urge you to check out what we have. And then the summer workshop, so 2 in-person educational events that we host are these CRISPR workshops to teach people how to do gene editing, and one of them is geared towards active scientists who can bring this knowledge back to their home lab. And the other one we just started, again with the help of Ariana Hirsch. This is for undergrads at Berkeley who are all really excited about CRISPR and wanted to do it themselves. And then in another do-it-yourself vein, we're working with the Lawrence Hall of Science and Adgene to develop an educational kit that people can order and take to their, or teachers can order specifically, and do in their classrooms. So there was a comment at CRISPRCon from someone that was like, if you want people to be interested in science, have these kids change bacteria from one color to another, have them do it in their classroom, and then they'll come home and tell their parents about it, and they'll see that excitement. And so we're trying to make that happen, and we're partway through developing that. So any of these things that you have any comments on or feedback, I'd be happy to hear it later, and if you have ideas of your own, we're really always looking for people to volunteer to give a tour of your lab, or do Ask a Scientist, or anything you can dream up. So yeah, definitely let me know if you have any ideas. And I guess with that, Jennifer will talk.
I want to let everyone get outside and have a look at the posters and write on the tables and all of that, but I just want to point out, I want to close by pointing out that none of this could happen without incredible support. So in addition to all of the folks that you've heard from and the people that have been mentioned, I'd like to also give a big shout out to Diane Ferris, who's standing over here, who does administrative support for the IGI and so many other things. Also Melanie Cantarutti, I don't know if she's in the room or not, but she's works in the Doudna lab, but she's been a very close partner with the IGI and has helped with a lot of the events that we've done. I also want to thank Letty Light, who is and Mara McClellan, who's not in the room, but Letty, I think, is here somewhere. Letty is working on development, so she's been instrumental in helping the IGI to attract the kind of resources that we need to put together these sorts of programs and be able to fund research and put out calls for proposals and things like that. And our two funders right now, we've had very generous, we've had two major donors that have given money, and we've had a number of other people that have given smaller amounts of money. We're really grateful to our donors, and we're actively working on additional donations. And finally, couldn't do anything at all without great scientific colleagues. In addition to all of the folks that you've heard from, and you didn't hear from Bruce or Jonathan, but they've been really integral to a lot of the things that we're doing, especially at UCSF. Keith Yamamoto at UCSF was a big player in the early days of getting the IGI founded, as well as colleagues here at Berkeley, including Bob Tijan, Ed Penhote, and Mike Botchin. So huge thanks to all of them as well. And with that, I'd like to thank you for coming, and I hope you enjoy the afternoon.